Hello everyone, I am Ananya Chatterjee. I am a PhD student in IISc Bangalore. I am one of the TAs of the course Introduction to Cell Biology, Cell Components, Organization and Processes. So the course instructor is Professor Shikha Larolaya from IISc. So today we'll be discussing about week 4 contents and some previous year question answers. I hope my screen is visible and my voice is audible as well to you. So we will be discussing week 3's assignments, or I think this is week 4's assignments. So the first question was, which of the following kinases is not required for mitotic events to take place? So when we will discuss about different kinases which are required for mitotic events, we will see that there are different kinases like polo-like kinase that helps in assembly of the bipolar mitotic spindles. We know mitotic spindles, so they help in assembly bipolar mitotic field. Bipolar means they have two sides, positive and negative. It phosphorylates proteins required for separation of the spindle poles early in the mitosis. So this polo-like kinase, as the name suggests, it also phosphorylates proteins that are required to separation of the spindle poles. Then comes Aurora kinase, Aurora A. It helps in assembly and stability of spindle. Aurora kinase or Aurora B, it helps in attachment of sister chromatids to the spindle. So we know that during mitosis we need to have the sister chromatid attached with the spindle. So with that respect, Aurora kinase B plays a role. So here they ask that which is not required. So the answer will be none of the above, because all three are required. The key event that occurs at the metaphase to anaphase transition is sister chromatid dissociation and segregation, sister chromatid association on the equatorial plane, sister chromatid condensation, all of the above. So the key event that occurs at the metaphase to anaphase transition. So what happens when a cell goes from metaphase to anaphase? So in metaphase, the chromatids get segregated into, like in metaphase what happens, the chromatids lie at the middle of the cell, and in anaphase what happens, the chromatids get segregated into two poles. So what will happen? Sister chromatid dissociation and segregation, sister chromatid association and condensation. So association will happen when the cell will be in metaphase. Okay, when the cell is going to anaphase state, the sister chromatid will dissociate and segregate. So the answer will be this. And sister chromatin condensation happens during replication or during the S phase. Then the question is, sister chromatid cohesion is established in G1 phase, S phase, metaphase and anaphase. So the cohesion will discuss, it occurs or it is established in S phase. So how now cohesions hold sister chromatids together. So what is the role of cohesins? It holds the sister chromatids. We know sister chromatids. Let's see. So they hold the sister chromatids together. So what happens when the cell is at S phase? Each replicated chromosome consists of a pair of identical sister chromatids, okay, and that are glued together along their length. This sister chromatid cohesion sets the stage for a successful mitosis because it greatly facilitates the attachment of the two sister chromatids to opposite poles of the mitotic spindle. So you can imagine that how to achieve this bipolar attachment if sister chromatids were allowed to drift apart after S phase. Indeed, defects in sister chromatid cohesion in yeast mutants, for example, lead inevitably major errors in chromosome segregation. So sister chromatid cohesion depends on a large protein complex called cohesin, which is deposited at many locations along the length of each sister chromatid as the DNA is replicated in S phase. Two of the subunits of cohesin are members of a large family of proteins called SMC proteins for structural maintenance of chromosomes. So cohesin forms a giant ring-like structure, and it has been proposed that this surround the two sister chromatids, okay. So it surrounds the two sister chromatids. Sister chromatid cohesion also results, at least in part, from DNA catenation, the intertwining of sister DNA molecules that occur when two replication forks meet during DNA synthesis. The energy topoisomerase 2 gradually disentangles the catenated sister DNAs between S phase and early mitosis by cutting one DNA molecule and passing the other through the break, and then again they reseal the cut DNA. And when the catenation happens, the sister chromatid cohesion depends primarily on cohesion complex. So the sudden and synchronous loss of sister cohesion at the metaphase to anaphase transition therefore depends primarily on disruption of these complexes. So this is the SMC molecule in this protein. You can see this is C terminal, N terminal. This is basically ATPase domain. It has a hinge. So the proteins are what SMC molecules are consist of: SMC3, SCC3, SCC1, SMC1. There is a hinge, you can see. So they hold the chromatids like that. So it is a protein complex with 4 subunit. These 4. So these two subunit SMC1 and SMC3 are coil proteins with so they have a ATPase domain, and the additional two subunits SCC1 and SCC3, they connect the ATPase head domains, forming a ring structure that may encircle the sister chromatid as shown in C. The ATPase domain are required. These ATPase domain are required for cohesion leading on the DNA. So now the next question is, which of the following class of proteins are not present in the pericentriolar material? Cell cycle regulators, signaling molecules, microtubule organizers, none of the above. So pericentral materials refers to the proteinaceous material that surround the centriole. Two small microtubule based cylinders, and with them constitute the centrosome, the main microtubule organizing center. So this which of the protein are not present? So none of the above will be the answer, because the class of proteins are not present. So here everything will be present to make the system run. So the answer will be none of the above. The next question is, microtubules that overlap with each other when in anti-parallel orientation are called astral microtubules, kinetochal microtubules, interpolar microtubules, intrapolar microtubules. So if you see the orientation, these are centrosomes. So from the microtubule organizers, the microtubule will arise in the fashion of -2 plus and the direction. So if you see, the interpolar microtubules are at the very this side. So when -2 placent is going from one centrosome to other to the middle of the cell, and to the from the another centrosome, they are also coming. So when we can see there is an anti-parallel orientation of microtubules that overlap, that kind of thing is called interpolar. So there is another kind of microtubules which is generating from negative to positive and going to the cell periphery, that is astral microtubule. And another is non-kinetoconal microtubules which is going here to here, but it is not overlapping with any anti-parallel oriented microtubules. Which of the following proteins is absolutely required for metaphase to anaphase transition? So what happens when we go from metaphase to anaphase? So we need to separate two sister chromatids, right? So for separating two sister chromatid, we need a protein that helps in that separation and dissolve this cohesion interaction between two sister chromatids. So for that, we know that separase is a protein that is required. So what it does, separase is recruited to the mitotic chromosomes to dissolve sister chromatid cohesion in DNA dependent manner. The spindle pole body present in budding yeast is equivalent to centrosome, lysosome, Golgi body, ear membrane. So the functional equivalent of the centrosome is in budding yeast is called spindle pole body. So it lies embedded in the nuclear membrane which remains intact during mitosis. The spindle pole body is a cylindrical multi-layered organelle with the most visible layers being the outer and the central plex. So you have to know that the spindle pole body, the spindle pole body is also can be depicted as centrosome in the case of yeast. The Frapp experiment on microtubules yields which of the following inferences? Individual microtubules are dynamic and their subunits are constantly being replaced. Individual microtubules are constantly polymerizing at both ends. Tubulin remains at the equatorial plate of the cell cycle. Microtubules are static and stable in nature. So, in the class lecture you can see the Frapp experiment which on the microtubule leads. It depicts basically that microtubules are very dynamic because tubulin proteins are going and coming out and they are constantly being replaced. So if you do some kind of Frapp experiment, you can see that they are very dynamic and they are going out and going in. So the answer will be the Frapp experiment can give the result of dynamicity in the case of that experiment. So the next question is, select the correct statement regarding spindle attachment to the chromosome. The option one is sister kinetochores are assembled in back-to-back orientation to reduce the chance of mono-orientation. Option 2, cohesion at the centromere makes the paired sister chromatids to be more fluid in nature. Biorientation of chromosomes is important to generate tension at the kinetochore between the sister chromatids. So you have to choose correct statement regarding the attachment to the chromosome. So when you see sister kinetochores are assembled in back-to-back orientation to reduce the chance of mono orientation, that is correct. So when a sister chromatid is there, so the kinetochore assembles a back-to-back to reduce the chance of mono orientation. They don't, we'll discuss in next slides, they don't place and a mono oriented. Then the second option, cohesion at the centromere makes the paired sister chromatids to be more fluid in nature. No, it's not right. It makes it more rigid. And the third is biorientation of chromosomes is important to generate tension. Yeah, when biorientation of chromosome happens, it's important to generate tension at the kinetochore. So if you pull like that, it will create a tension and between two sister chromatids. So the answer will be 1 and 3. The last question was, extended binding of NET1 to CDC14 can lead to enhance activity of CDC14, delay in mitosis exit, early entry into cytokinesis, none of the above. So for understanding this, you have to know what is the role of NET1, CDC2 and so on. So if you see mitotic exit network, so what happens, so this is a protein TEM on GTP, it has a GTPase activity. So if so, what happens, from TEM on GTP, under GTPase activity you can go to TEM on GTP. So and here some other proteins are associated. So in this case you can see in the kinase cascade, this NET1 is inhibiting CDC14, which is a phosphatase, and who CDC14 helps in mitotic exit. So if NET1 binds with CDC14, so the effect of phosphatase of CDC14 is getting hindered. So then what will be delay? The delay will be in mitosis exit. So today we'll just brush up some concepts of mitosis, which is being discussed in the week 4 lecture contents. So we know that when, so yesterday's class we have gone through S phase, the interphase, all the interphase phases and all. So so after completion of S phase and when cells transits to G2, so then the cell undergoes the dramatic upheaval of M phase. So what happens, there is the cell starts preparing itself for M phase. So this begins with mitosis, during which the sister chromatids are separated and distributed to a pair of identical daughter nuclei, each with its own copy of genome. Mitosis is traditionally divided into 5 stages: prophase, metaphase, prophase, prometaphase, metaphase, anaphase and telophase, defined primarily on the basis of chromosome behavior as seen in microscope. As mitosis is completed, the second major event of M phase, cytokinesis. So from a regulatory point of view, mitosis can be divided into 2 major parts, each governed by distinct components of the cell cycle control system. First, an abrupt increase in mCDK activity. So mCDK helps in the cell to promoting it into mitosis. So at the G2M transition, there will be an increase in mCDK activity that triggers the events of early mitosis, prophase, prometaphase and metaphase. During this period, mCDK and several others mitotic protein kinase phosphorylate a variety of proteins, leading to the assembly of the mitotic spindle and its attachment to the sister chromatic pairs. The second major part of mitosis begins extraction of securin, liberating a proteases that cleaves cohesin and thereby initiates separation of sister chromatids. So there is another major part, what happens during the metaphase to anaphase transition. What happens when in the metaphase sister chromatids are attached with each other. So we have to dissolve this bond and make the chromatids free so that they can go to 2 different poles in the cell. So APCC also promotes the destruction of cyclins, which leads to CDK inhibition, inactivation and dephosphorylation of CDK targets, which is required for all events of late M phase, including the completion of anaphase. So the completion of anaphase means the disassembly of mitotic spindle and the division of the cell by cytokinesis. In the cytokinesis, what happens? The cell is getting divided into 2 halves, each with an identical nucleus. So how mCDK drives entry into mitosis? So one of the most important feature of cell cycle control is a single protein kinase mCDK. Brings about all of the drivers and complex cell rearrangements that occur in the early stages of mitosis. At a minimum, mCDK must induce the assembly of the mitotic spindle and ensure that each sister chromatid in a pair is attached to the opposite pole of the spindle. So it also triggers chromosome condensation, the large-scale reorganization of the intertwined sister chromatids into compact rod-like structures. So in animal cell, mCDK, what it promotes, the breakdown of nuclear envelope. So whatever you have to understand it, you have to understand what is happening in the cell cycle in each stage: metaphase, prometaphase, metaphase, anaphase, telophase, like that. So MCDK promotes, so at first you have to break down the nuclear envelope so that the genome comes out, and then they condense, and like that. So then you have to also rearrangement the actin cytoskeleton and Golgi apparatus. So every process is thought to be initiated when MCDK phosphorylates specific proteins involved in the process. Although most of these proteins have not yet been identified, MCDK does not act alone to phosphorylate key proteins involved in early mitosis. Two additional families of protein kinase, the polo-like kinase and aurora kinase, also make important contribution to the control of early mitotic events. So the polo-like kinase, what it does, it is required for the normal assembly of a bipolar mitotic spindle, in part because it phosphorylates proteins involved in separation of the spindle poles early in mitosis. The aurora kinase, aurora A, also helps control proteins that govern the assembly and stability of the spindle, whereas aurora B controls attachment of sister chromatids to the spindle, and we'll discuss it later. So what happens, another scenario, dephosphorylation activates MCDK at the onset of mitosis. How MCDK is getting activated? So MCDK activation begins with the accumulation of M cyclin. This is also called as cyclin B in vertebrate cells. In embryonic cell cycles, the synthesis of M cyclin is constant throughout the cell cycle, and the M cyclin accumulation results from the high stability of the protein interface. In most cell types, however, M cyclin synthesis increases during G2 and M, owing primarily to an increase in M cyclin gene transcription. The increase in M cyclin protein leads to a corresponding accumulation of MCDK, and there will be a complex of CDK1 and M cyclin, and so on. So in this picture we can see CDK1 and M cyclin gets attached, and they will make a complex, and but this is inactive MCDK. When CDK activated kinase comes and it phosphorylates it, so then that thing will be active. But along with that, CDK inhibitory kinase V1 is there, which can also phosphorylate at a inhibitory site. So if it also phosphorylate, what happens then? It will become inactive. Then there is a phosphatase CDC25. It comes and then dephosphorylate the inhibitory phosphate. So it makes the thing as active MCDK. So the activation of MCDK, CDK1 associates with M cyclin. As the levels of M cyclin gradually rise, the resulting MCDK complex is phosphorylated on an activating site by the CDK activating kinase, and on a pair of inhibitory sites by the V1 kinase. So the resulting inactive MCDK complex is then activated at the end of G2 by the phosphatase CDC25. CDC25 is further stimulated by active MCDK. Active MCDK resulting in positive feedback. So as we were discussing about cohesine, we have to also know what is condensing, what makes that genome very condensed. So what happens when at the end of S phase? We know that DNA replication happens during S phase. So at the end of S phase, the DNA molecules is immensely long. So if we stretch it out, it will be in fit. So this immensely long DNA molecule should be condensed. So these molecules, DNA molecules of the sister chromatids, are tangled in a mass of partially catenated DNA and proteins. Any attempt to pull the sisters apart in this state would undoubtedly lead to breaks in the chromosomes. To avoid this disaster, the cell devotes a great deal of energy in early mitosis to gradually reorganizing the sister chromatids into relatively short, distinct structures that can be pulled apart more easily in anaphase. These chromosomal changes involve two processes: chromosome condensation, in which the chromatids are dramatically compacted, and sister chromatid resolution, whereby the two sisters are resolved into distinct separable units. Resolution results from the decatenation of the sister DNAs accompanied by the partial removal of cohesion molecule along the chromosome arms. As a result, when the cell reaches metaphase, the sister chromatid appear in the microscope as compact rod-like structures that are joined tightly at their centromeric regions and only loosely along their arms. So this is a proposed mechanism of condensing. It holds sister chromatids like that, and then it entangles. So the condensation and resolution of sister chromatids depend, at least in part, on a 5-7-8 protein complex called condensin. So this structure is related to cohesin complex that holds sister chromatids together. So it contains two SMC subunits like cohesin. So you can find a similarity between condensin and cohesin. So along with two subunits, it also has three non-SMC subunits. So cohesin may form a ring-like structure that somehow uses the energy provided by ATP hydrolysis to promote the compaction and resolution of sister chromatids. Condensin is able to change the coiling of DNA molecules in a test tube, and this coiling activity is thought to be important for chromosome condensation during mitosis. And interestingly, phosphorylation of condensin subunits by mCDK stimulates this coiling activity, providing one mechanism by which mCDK may promote chromosome restructuring in early mitosis. So here a brief overview of different phases of M phase. So in prophase, what happened? There will be you can see this is an intact nuclear envelope. These are two centrosomes. So from there mitotic spindles are emerging out. This is kinetochore holding the sister chromatids. So these are condensing replicated chromosome consisting of two sister chromatids held together along their length. So at prophase, what happens? The replicated chromosomes, each consisting of two closely associated sister chromatids, condense. Outside the nucleus, the mitotic spindle assembles between the two centrosomes, which have replicated and moved apart. For simplicity, only 3 chromosomes are shown here. So in diploid cells, there will be 2 copies of each chromosome present in the fluorescence microscope. Fluorescence micrograph you can see. So this is a real image. So here the chromosomes are stained orange and the microtubules are stained green. In prometaphase, what happened? The nuclear, you have to then break this nuclear envelope. So there will be fragmentation of nuclear envelope. Then centrosome will be at 2 pole or 2 spindle pole. This is kinetochore microtubule chromosomes in active motion. So prometaphase starts abruptly with the breakdown of nuclear envelope. Chromosomes can now attach to spindle microtubules via their kinetochores and undergo active movement. In metaphase, then what happens? The chromatids will be at the equatorial position. So you can see centrosome at spindle pole. These are kinetic microtubule. At metaphase, the chromosomes will be aligned at the equator of the spindle, midway between the spindle poles. The kinetochore microtubules attach sister chromatids to opposite poles of the spindle. In anaphase, now the sister chromatids will be separated. So the sister chromatids synchronously separate to form 2 daughter chromosomes, and each is pulled slowly towards the spindle pole at its faces. The kinetochore microtubules get shorter and the spindle poles also move apart. Both processes contribute to chromosome segregation. So here we can see shortening kinetochore microtubule. Those are spindle pole moving outward. Those are 2 daughter chromosomes. Here is a real view. You can see sister chromatids are getting separated with the help of microtubules. In telophase, what will happen? So the two sets of daughter chromosomes arrive at the poles, at two poles of the spindle, and they will now decondense. A new nuclear envelope will reassemble around each set, completing the formation of two nuclei and marking the end of mitosis. The division of the cytoplasm begins with contraction of the contractile ring. So these are interpolar microtubule centrosome. This is nuclear envelope just starting making. This is contractile ring starting to contract the cell to divide to help in cytokinesis. And this is a daughter chromosome at two spindle pole. Now there will be cytokinesis or the cell division. So in this case, this contractile ring, they are creating cleavage furrow, and which will eventually help in dividing these two cells, dividing this one cell into two cells. So during cytokinesis, the cytoplasm is divided in two by a contractile ring of actin and myosin filaments, which pinches the cell in two to create two daughters, each with one nucleus. So this is reformation of interphase array of microtubules nucleated by the centrosome. So now we are talking about mitotic spindles. So what are they? The mitotic spindle is a microtubule machine, microtubule based machine. So we know that the cytoskeleton is made up of three components: microtubules, microfilaments, and intermediate filaments. So what are microtubule based machine? The central event of mitosis, that is chromosome segregation, depends in all eukaryote on a complex and beautiful machine called the mitotic spindle. The spindle is a bipolar array of microtubules which pulls sister chromatid apart in anaphase, thereby segregating the two sets of chromosomes to opposite ends of the cell, where they are packaged into daughter nuclei. MCDK triggers the assembly of the spindle early in mitosis in parallel with the chromosome restructuring just described. So before we consider how the spindle assembly and how its microtubule attaches to spindle chromatids, we have to understand the basic feature of spindle structure. So the mitotic spindle is a bipolar array of microtubules. So it has a plus end and minus end. So the minus ends of which are focused at the two spindle poles, and the plus ends which radiate outside the poles. So the plus ends of some microtubules called the interpolar microtubules overlap with the plus ends of another microtubule, resulting in an anti-parallel array. And the plus ends of other microtubules, the kinetochore microtubules, are attached to sister chromatid pairs at large protein structures called kinetochores, which are located at the centromere of each sister chromatids. Finally, many spindles also contain astral microtubules that radiate outward from the poles and contact the cell cortex, helping to position the spindle in the cell. So here is the diagrammatic representation of diagrammatic representation of the metaphase mitotic spindle in an animal cell. So these are two centrosomes at two poles. This is also called spindle poles. Here microtubules are radiating from negative to positive. So this which are radiating and going outside or going towards the peripheral sides are called astral microtubules. Those blue colored are kinetochores microtubules because they are associated with the kinetochores of sister chromatids. These red ones are interpolar microtubules. So here you can see these red ones are crossing each other in an anti-parallel way. So from one pole this is one in interpolar microtubules, from the other pole this is another. So this is going from negative to positive. And these are some motor proteins. So microtubule dependent motor proteins govern spindle assembly and function. The function of the mitotic spindle depends on numerous microtubule dependent motor proteins. These proteins belong to two families: the kinesin related proteins, which usually move toward the plus end of microtubules, and dyneins, which move toward the minus end. In the mitotic spindle, these motor proteins generally operate at or near the ends of microtubules. Four major types of motor proteins, kinesin 5, kinesin 14, kinesin 410, dynein, are particularly important in spindle assembly and function. Kinesin 5, what it does, it contains two motor domains that interact with the plus ends of antiparallel microtubules in the spindle midzone. So because the two motor domains move toward the plus ends of microtubules, they slide the two antiparallel microtubules past each other towards the spindle poles, pushing the poles apart. Kinesin 14 proteins, by contrast, are minus end directed motors with a single motor domain and the other domains that can interact with a neighboring microtubule. They can cross-link antiparallel interpolar microtubules at the spindle mid-zone and tend to pull the poles together. What is the function of kinesin 4 and 10? They are also called chromokinesin, and those are plus end directed motors that associated with chromosome arms and push the attached chromosome away from the pole or the pole away from the chromosome. And what is the function of dyneins? Dyneins are minus end directed motors that together with associated proteins they also organize microtubules at various locations in the cell. So they basically link the plus ends of astral microtubules to components of the actin cytoskeleton at the cell cortex. For example, by moving toward the minus end of the microtubules, the dynein motors pull the spindle poles toward the cell cortex and away from each other. So this is again the diagrammatic representation what I was telling. So these major motor proteins you have to remember and their function. So major motor proteins of the spindles are dynein and kinesins. In the kinesins there are also several types: kinesin 14, kinesin 5, kinesin 4, 10. So four major classes are shown here. So dynein is helping going towards this direction, kinesin 4 is going towards this direction, and kinesin 5 can go in both direction, from negative to positive, from like this. And so yeah, and yes, so these are interpolar. So as these are interpolar, you can see, so if it can work through two both the interpolar microtubules, and these are also dynein going from positive to negative. So regarding the kinesin 5, so it contains two motor domains so that it can interact with the plus ends of the antiparallel microtubules in the spindle mid-zone. So because these two motor domain move toward the plus ends of the microtubule, they slide the two antiparallel microtubules, so they lie like that. So they can slide the two antiparallel microtubules past each other towards the spindle pole. So kinesin 5 is important in the respect of interpolar microtubules. So they are also plus directed in both the microtubules. So now we'll discuss a little bit regarding centrosome duplication occurs early in the cell cycle. So most animal cells contain a single centrosome that nucleates most of the cell cytoplasmic microtubules that we have seen. So this centrosome duplicate when the cell enters the cell cycle, so that by the time cell reaches mitosis, there are two centrosomes. Centrosome duplication begins at about the same time as the cell enters the S phase. The G1S CDK, a complex of cyclin E and CDK2 in animal cells that triggers cell cycle entry, that also helps initiate centrosome duplication. So G1S CDK also helps in centrosome duplication. So these two centrioles in the centrosome separate, and each nucleates the formation of single new centriole, and resulting in two centriole pairs within a enlarged pericentriolar matrix. So this centrosome pair remains together on one side of the nucleus until the cell enters mitosis. There are interesting parallels between centrosome duplication and chromosome duplication. Both use a semi-conservative mechanism of duplication in which the two halves separate and serve as template for construction of a new half. Centrosomes, like chromosome, must replicate once and only once per cell cycle to ensure that cell enters mitosis only with two copies. An incorrect number of centrosomes could lead to deflect in spindle assembly and thus errors in chromosome segregation. So we have to ensure that there are only two copies of centrosome before it is going to M phase or mitotic phase. So here the centriole replication looks like. So the centrosome consists of centriole pair and some associated pericentriole matrix. At a certain point G1, the two centrioles of the pair separate by some few micrometer. They get separated. During S phase, the daughter centriole begins to grow near the base of each mother centriole. So another daughter centriole begins to grow and at a right angle to it. The elongation of the daughter centriole is usually completed in G2. The two centriole pairs remain close together in a single centrosomal complex until the beginning of M phase, when the complex splits into two and the two daughter centrosomes begin to separate. Each centrosome now nucleates its own radial array of microtubules, this green rays, and they start helping in the cell division. Now coming to the role of MCDK into initial spindle assembly. So spindle assembly begins in early mitosis when the two centrosomes move apart along the nuclear envelope. So pulled by dynein motor proteins that link astral microtubules to the cell cortex. The plasends of microtubules begin the centrosomes interdigitate to form the interpolar microtubules, and kinesin 5 motor proteins associate with these microtubules and push the centrosomes apart. Also in early mitosis, the number of gamma tubulin ring complexes in each centrosome increases greatly, increasing the ability of the centrosome to nucleate new microtubules, a process called centrosome maturation. The balance of opposing forces generated by different types of motor proteins determines the final length of spindle. Dynein and kinesin 5 motors generally promote centrosome separation and increase spindle length. Kinesin 14 proteins do not do the opposite. They tend to pull the poles together. It's not clear how the cell regulates the balance of opposing forces to generate the appropriate spindle length. So here we'll discuss about MCDK, the role of MCDK and other mitotic protein kinase. Those are required for centrosome separation. So they basically ensure that those two centrosomes will separate and then maturate, and MCDK and Aurora phosphorylate kinesin 5 motors and stimulate them to drive centrosome separation. Aurora A PLK also phosphorylates components of the centrosome and thereby promotes the maturation. So this maturation is happened during prophase, and MCDK plays a major role into it. Now microtubule instability increases greatly in mitosis. So most of the animal cells in interphase contain a cytoplasmic array of microtubules, and those are radiating from the single centrosome. The microtubules of this interphase array are in a state of dynamic instability, in which individual microtubules are either growing or shrinking and stochastically switch between the two states. So the switch from the growth to shrinkage is called catastrophe. So the dynamics of microtubules will discuss more during the cytoskeleton class. So the switch is also called catastrophe, and the switch from shrinkage to growth is called rescue. New microtubules are continually being created to balance the loss of those that has been disappeared completely by depolarization. So entry into mitosis signal an abrupt change in its cell's microtubules. The interface array of few long microtubules radiating from the single centrosome is converted to a larger number of shorter and more dynamic microtubules emanating from both centrosome. During prophase and particularly in prometaphase and metaphase, the half-life of microtubules decreases dramatically. This increase in microtubules instability coupled with the increased ability of centrosome to nucleate microtubule, as we have talked earlier, results in remarkably dense and dynamic arrays of spindle microtubules that are ideally suited for capturing sister chromatids. Microtubule dynamics are controlled in the cell by a variety of regulatory proteins, including microtubule associated proteins that promote stability and catastrophe factors that destabilize microtubule placents. Changes in the activity of these regulatory proteins are responsible for the changes in microtubule dynamics that occurs in mitosis, and many of these changes result in the phosphorylation of specific proteins by MCDK and other mitotic protein kinase. So here we will see the spindle self-organization by motor proteins. So mitotic chromosomes stimulate the local activation of proteins that nucleate and promote the formation of microtubules in the vicinity of the chromosome. Kinesin 5 motor proteins organize these microtubules into antiparallel bundles. So kinesin 5 are there, they organize this. So they have, as I was discussing, that they have two motor domains. So they can interact with both the microtubules. So they are organizing this antiparallel bundles, positive to negative and negative to positive. So it's called antiparallel. While placent directed kinesin 4, what they do, so 4 and 10 both, they link the microtubules to chromosome arms. So they link the microtubules to the chromosome arms and push minus ends away from the chromosome. Okay, so they push minus end away from the chromosome. And dynein and kinesin 14 motors, together with numerous other proteins, they focus on minus ends into a pair of spindle poles. So you can see in the minus 4, they are forming a pair of spindle poles and they remain there. So this is what happens. Nucleation happens, then antiparallel cross-linking by kinesin 5. Outward push happens by kinesin 4 and 10. They pushed outward this negative end, and then focusing of poles, they basically what they do, what is focusing, you can see that they're going to a single point there. So this is kind of a focus. So they focus these poles into two poles. So dynein and kinesin 14 helps here. So now we are talking about kinetochore. How does kinetochore look and what is kinetochore? So in this fluorescence microscope, you can see metaphase chromosome stained with a DNA binding fluorescent dye and with human autoantibodies which can react with kinetochore proteins. So the two kinetochores, one associated with each sister chromatid, are stained as red. B, this is a drawing of metaphase chromosome showing its two sister chromatids attached to the placents of kinetochore microtubules. Each kinetochore forms a plaque on the surface of the centromere. This is the electron micrograph of an anaphase chromatid with microtubules attached to its kinetochore. While most kinetochores have a trilaminar structure, the one shown here from a green algae has an unusual complex structure with additional layer. So this is anaphase chromatic, this is kinetochore, this is a direction of chromatid movement. These are microtubules which are embedded in the kinetochore. This is the electron micrograph image. Now we'll briefly go through chromosome attachment to the mitotic spindle in animal cells. So this is late prophase, this is early prometaphase where now chromatids are going towards equatorial pole. This is mid prometaphase and this is metaphase where these are oriented as like by orientation, you can see. So in late prophase, what happens, the mitotic spindle poles have moved to opposite ends of the nuclear envelope with an array of overlapping microtubules between them. Following that, a nuclear envelope breakdown, the sister chromatid pairs are exposed to the large number of dynamic plus ends of microtubule radiating from the spindle poles. So in most cases, the kinetochores are first attached to the sides of this microtubules, while at the same time the arms of the chromosomes are pushed outward from the spindle interior, preventing the arms from blocking microtubule access so that it can access the kinetochore. Eventually, the laterally attached sister chromatids are arranged in a ring around the outside of the spindle. Most of the microtubules are concentrated in this ring so that the spindle is relatively hollow inside. Dynamic microtubule plus ends eventually encounter the kinetochores in an end-on in orientation and are captured and stabilized. Stable end-on attachment to both poles result in biorientation. So now this sister chromatids are stable and this results in biorientation. So kinetochores are attaching at two sides and centrosomes are away. Inside you can see those microtubules are helping in arranging these chromatids. So how the kinetochore is getting attached with the microtubule? This can attach like this can attach like this or this. So alternative forms of kinetochore attachment to the spindle poles. So if from the one pole only microtubule is getting attached with the kinetochore, this is unstable. So initially a single microtubule from a spindle pole binds to one kinetochore in a sister chromatid pair, and kinetochore in a sister chromatid. So additionally microtubules can then bind to the chromosome in various ways. So in this case, a microtubule from the same spindle can attach to the other kinetochores, but you can imagine that this also unstable. This can pull the chromatid towards a certain pole. Another thing can happen where microtubules from both the spindle pole are attaching with the same kinetochore. This is also tremendous unstable. And third option is that from the two pole microtubule will attach to other kinetochore, so that it will create a very stable tensile force. So this is what happens during metaphase to anaphase. This is all about. Now we'll see how APCC helps in metaphase to anaphase transition. So the initiation of sister chromatid separation by APCC. So here you can see this APCC complex is inactive. When CDC20 binds it, it becomes active. So what is the role of it? We know that when securin binds with inactive separase, that the separase becomes inactive. Okay, so what it does, it helps in ubiquitination and degradation of securin, so that the separase is now released and it becomes active. Eventually what happens, mCDK, this is condensin protein. So what happens when it helps in mCDK helps in metaphase and the sister chromatids are now at the mitotic spindles, this separase comes into play and it breaks these cohesins which are holding those two sister chromatids. So those clipped and dissociated cohesins, and you can see this is the anaphase. Now if sister chromatids is breaking, so then two chromatids will go at two poles. So chromosome segregation will occur in anaphase A and B. So the certain loss of sister chromatid cohesion at the onset of anaphase leads to sister chromatid separation, which allows the forces of the mitotic spindle to pull the sisters to opposite poles of the cell, called chromosome segregation. The chromosomes move by two independent and overlapping processes. The first, anaphase A, is the initial poleward movement of the chromosome, which is accompanied by shortening of the kinetochore microtubules. The second, anaphase B, is the separation of the spindle poles themselves, which begins after the sister chromatids have separated and the daughter chromosomes have moved some distance apart. So this chromosome movement in anaphase A depends on a combination of two major poleward forces. The first is force generated by the microtubule depolymerization at the kinetochore, which results in the loss of tubulin subunits at the plus end as the kinetochore moves towards the pole. The second is provided by the microtubule flask, which is the poleward movement of the microtubules towards the spindle pole, where minus end depolymerization occurs. The relative importance of these two forces during anaphase varies in different cell types. In embryonic cells, the chromosome movement depends mainly on microtubule flask, for example, whereas movement in yeast and vertebrate somatic cells results primarily from forces generated at the kinetochores. So, although sister chromatid separation initiates the chromosome movements of anaphase A, other mechanisms also ensure correct chromosomes movements in anaphase A and spindle elongation in anaphase B. So, most importantly, the completion of a normal anaphase depends on the dephosphorylation of CDK substrates, which in most cells results from the APCC dependent destruction of cyclins. So, if M-cyclin distraction is prevented by the production of a mutant form that is not recognized by the APCC, for example, sister chromatid separation generally occurs, but the chromosome movements and the microtubule behavior of anaphase are abnormal. The relative contributions of anaphase A and B to chromosome segregation vary greatly depending on the cell type. In mammalian cell, anaphase B begins shortly after anaphase A and stops when the spindle is about twice in metaphase length. So in contrast, the spindles of yeast and certain protozoa primarily use anaphase B to separate the chromosomes at anaphase, and their spindles elongate to up to 15 times their metaphase length. So you can see this anaphase A and anaphase B. So here force are generally because of the kinetochores, and here is forces generally because of the microtubules. So basically the chromatids are getting separated from each other and going towards the two poles. So now eventually after chromatid segregation at the anaphase, the cell has to start cytokinesis. The final step in the cell cycle is cytokinesis, the division of the cytoplasm in two. In most cells, cytokinesis follows every mitosis, although some cells such as early Drosophila embryos and some mammalian hepatocytes, heart muscle cells undergo mitosis without cytokinesis and thereby acquire multiple nuclei. In most animal cells, cytokinesis begins in anaphase and ends shortly after the completion of mitosis in telophase. So the first visible change of cytokinesis in an animal cell is sudden appearance of a pucker or a cleavage furrow on the cell surface, and the furrow rapidly deepens and spreads around the cell until it completely divides the cell in two. The structure underlying this process is a contractile ring, a dynamic assembly composed of actin filaments, myosin 2 filaments, and many structural and regulatory proteins. So during anaphase, the ring assembles just beneath the plasma membrane. The ring gradually contracts, and at the same time, fusion of intracellular vesicles with the plasma membrane inserts new membrane adjacent to the ring. So this addition of membrane compensates for the increase in surface area that accompanies cytoplasmic division. When the ring contraction is completed, membrane insertion and fusion fill the gap between the daughter cell. So here comes the role of actin and myosin 2 in the contractile ring that generate the force for cytokinesis. So in interphase cell, what happens? Actin and myosin 2 filaments form a cortical network. So if you consider a plasma membrane, so actin and myosin forms a cortical network underneath the plasma membrane. In some cells, they also form large cytoplasmic bundles called stress fiber. So as cells enter mitosis, these arrays of actin and myosin disassemble. Much of the actin reorganizes and myosin 2 filaments are released. As the sister chromatins separate in anaphase, actin and myosin 2 begin to accumulate in the rapidly assembling contractile ring, which also contains numerous other proteins that provide structural support or assist in ring assembly. Assembly of the contractile ring results in part from the local formation of new actin filaments, which depends on forming proteins. So this is very details. We will discuss the detailed actin filament and myosin in cytoplasm class, cytoskeleton class. Now we'll just making you understand that what happens after anaphase and how the cell is dividing. So the overlapping array of actin and myosin 2 filament contract to generate the force that divides the cytoplasm in two, and this contraction begins and a ring exerts a force large enough to bend a fine glass needle, so that is inserted in the path. As a ring constrict, it maintains the same thickness, suggesting that its total volume and the number of filaments it contains decrease steadily. Moreover, unlike actin in muscle, the actin filaments in the rings are highly dynamic and their arrangement changes continually during cytokinesis. The contractile ring is finally dispensed with altogether when cleavage ends, as the plasma membrane of the cleavage furrow narrows to form the midbody. The midbody persists as a tether between the two daughter cells and contains the remains of the central spindle, a large protein structure derived from the antiparallel interpolar microtubules of the spindle midzone, packed tightly together within a dense matrix material. After the daughter cells separate completely, some of the components of the residual midbody often remain on the inside of the plasma membrane of each cell, where they may serve as a mark on the cortex that helps to orient the spindle in the subsequent cell division. So there is also a role of rho A in the regulation of the contractile ring during cytokinesis. So regulation of the contractile ring by the GTPase rho A. So rho A is also a GTPase. Like other rho family GTPase, rho A is activated by a rho GEF. Rho A is activated by a rho GEF. So what happens? It is rho A is now bound with GDP. If rho GEF comes and it phosphorylates GDP, it becomes GTP. So it is now active. So active rho A, what it does, the active GTP bound form of rho A is focused at the future cleavage site. By binding formins, activated rho A promotes the assembly of actin filaments in the contractile ring. So it binds with formin and actin filament. It helps in formation of or promotes in formation of actin filaments. By binding formins, activated assembly helps in the assembly, and by activating rho activated protein kinases such as ROCK, it stimulates myosin 2 filament formation and activity. So when rho A activates rho activated kinase including ROCK, it induces regulatory myosin light chain phosphorylation, and it subsequently promotes myosin 2 activation and moreover helps in assembly and contraction of actin myosin rings. So, three current models of how microtubules of the anaphase spindle generate signals that influence the positioning of the contractile ring. So, this is astral stimulation model, this is central spindle stimulation model, and this is astral relaxation model. So, no single model explains all the observation, and furrow positioning is probably determined by the combination of these three mechanisms, with the importance of the different mechanism varying in different organisms. So this is the three current models. So three current models of how microtubules of the anaphase spindle generates signals so that influence the positioning of the contractile ring. Now coming to cytokinesis. So these are plant cells. So the special feature of cytokinesis in a higher plant cell, The division plane is established before the M phase by a band of microtubules. So this is a band of microtubules and actin filaments. It is decided before the M phase only, where the cell is getting divided. At the beginning of telophase, after the chromosome have segregated and a new cell wall starts to assemble inside the cell at the equator of the old spindle, the interpolar microtubule of the mitotic spindle, remaining at the telophase form the phragmoplast. So the interpolar microtubules from the mitotic spindle that are remaining from the telophase, they can form phragmoplast, and the plasents of these microtubules no longer overlap but end at the cell equator. Golgi derived vesicles filled with cell wall material are transported along these microtubules and fused to form the new cell wall, which grows outward to reach the plasma membrane and original cell wall. The plasma membrane and the membrane surrounding the new cell wall now fuse, separating the two daughter cells. So this is the cycling levels. This is embryonic cell with no G1 phase. So you can see M cyclin level is high in M phase and then going down, and CDC20 APC activity will be higher when there will be transition from metaphase to anaphase. And this is cells with G1 phase. So there will be another activity of CDH1 in APCC activity keeps M cyclin level low. So you have to keep M cyclin level low during G1 phase. So there comes a role of this CDH1. So this is the creation of a G1 phase by stable CDK inhibition after mitosis. So in early embryonic cell cycle, CDC20 and APC activity rises at the end of metaphase, triggering M cyclin destruction. Because M CDK activity stimulates CDC20 APC activity, the loss of M cyclin leads to APC inactivation after mitosis. And in cells that have a G1 phase, the drop of M CDK activity in the late mitosis leads to the activation of CDH1 as well as accumulation of CDK inhibitor proteins.
APC stand antigen parenting cell.
No.
APC.
No. Anaphase promoting complex.
Okay.
Yeah. So this anaphase, what is the meaning tells you can understand. Anaphase promoting complex means that the cell is now going from metaphase to anaphase. Okay. So in cells that have a G1 phase, so here what happens when MCDK activity is getting down, that leads to the activation of another protein complex CDH1 and APCC. uh, this ensures a continued suppression of CDK activity after mitosis, and that is also required for the G1 phase. So week 5 6 contents are also out. I hope you are doing the assignments and submitting those assignments if you are registered for the course. And so this is a week 8 course. So until week 6 there are lectures already uploaded. Please go through the lectures and get back to us through discussion forum also, and in the class also, if you want to emphasize any specific topic. Currently I have just provided some slides which you may know a bit more, but along with that, if you want to emphasize on any particular topic, I can discuss with you. And in each class we discuss the assignments from the previous class. So if you have any doubt other than that, That also you can ask to us. Else we'll stop today's class and we'll meet next Tuesday again for discussing week 5 content, that is on meiosis, and we'll discuss some assignments again. Thank you everyone. Then we are stopping today's meeting. We'll meet next Tuesday again for discussing the course, and if you have any doubt, please let us know. Thank you.
Thank you, ma'am.